Hello everyone. Thank you for joining us today for the webinar. Before we begin, I would like to just tell you a few rules that we typically have during the webinar. First of all, you will be in the muted mode, but should be able to hear me and Josh. And for that, could you please type in the questions box whether you can hear me? Oh, yeah. Thank you. The second, the webinar is designed in the way that, so first we'll have an introduction, then Josh will present his slides, and then we'll have a Q&A session where you can ask the questions, and yeah, Josh will answer them. Also, the recording of the webinar will be available shortly after the presentation, and we'll share it with you, the link to our YouTube channel. For the new webinars and the announcements, please follow us on LinkedIn, Instagram, or Facebook. So a few words about Chemspace, and yeah, we're in 2021, but 2020 made a lot of difference on us, and I hope for you that was not only some things that you'll have a positive things to remember as well. And for Chemspace, that was really good year, and we were able to expand to more molecules that we can offer to our customers, and now we created the platform, a marketplace that can be a single place for ordering molecules from over 70 renowned suppliers. And with that, we hope we can help researchers to solve their problems. This catalog that we have contains over 1.6 billion molecules that are searchable online using just this simple form and pretty much conventional ways to search like exact match, substructure, or similarity. Also, you can do search by the just text search. For example, if you want to find aspirin or other molecules, you can type and find it. And also you can import the file. The platform is also, and the catalog is also available, as I said, online. It's available through API integration with client application, and it's available through punch-out site. The catalog, as I said, contains 1.6 billion molecules, out of which we have over 430,000 in-stock building blocks and over 6 million in-stock screening compounds provided by renowned suppliers. And along with that, we have billions of make-on-demand analogs to this in-stock compound that can be delivered pretty fast and reliably by our partners. With that, we have this, as I said, we have the really large catalog, and what we want to have it really useful for the customers and for the research community in general. And we have collaborations with companies like Molecular Forecaster, who can use our catalog and basically help their customers to resolve their problems. And with that, I would like to introduce you Joshua Pottel, President and CEO of Molecular Forecaster, who will give a talk on and describe the company and the services and the tools it provides. So Josh received his PhD from McGill University. He did a postdoc at UCSF as Brian Schoikert Lab, and he serves as president and CEO at Molecular Forecaster since 2016. So this is his title, and I think with that I would like to hand the word to Josh, and looking forward to hear his presentation. Josh.
Thank you, Yurii. Can everyone hear me? I think my thing is there. Let's show screen 2. Show my screen. Okay. Yurii, can you see the presentation?
Yes.
Okay. Do people prefer that I turn on my camera? I guess I'll just speak.
Yeah.
Okay, let's do this.
Yeah, we can see the slides.
There we go. Excellent. Okay, so thank you, Yurii, for inviting me to speak about what we do and talk a little bit about our partnership. Also, happy International Day of Women and Girls in Science. I found out that was today. Thanks to everyone for being here. I see we have a pretty good crowd, so I appreciate you all taking the time on Tuesday morning on the East Coast, wherever else, earlier on the Pacific side. I know we have some attendees and wherever else you may be. Thank you. I think Yurii pretty much touched on my background. I have training in some software development and in software application and drug design, being at McGill and then at UCSF. And I came back to Montreal in late 2018 to join Molecular Forecaster full-time and run the business development in the company. I'll tell you a little bit about us, and I'm going to kind of run through, I guess, the context of our company, some projects we've worked on, some of the software we develop, and how we might be able to help you in your research and your projects going forward. So our goal is really to be a self-sustaining leader in comp chem. We have expertise in drug discovery. You'll also see some expertise in chemistry. We're primarily chemists by training. So we do our own software development. We offer research services, as a CRO, and we also license our algorithms to some select partners around the world. How are we different than some of the other companies that exist? I'm sure you know plenty of computational chemistry companies. First, we lead with our high accuracy algorithms. These are developed primarily at McGill University, or at least it started that way. I'll talk a little bit more about what these algorithms can do and how they perform in various different circumstances. We act as a CRO using our own tools. I think that makes us pretty unique, offering our expertise in previous projects and things like that to companies in need. And lastly, try and make our software as easy to use as possible so that whether you're using the tools, in a complementary way while we're working on a project, or you want to use them in-house, you get to use these simple to use workflows that are kind of fully integrated with automation. It's set up with some point and click on a user interface to set up your problem, and then you can step away, go have coffee, go to sleep, whatever it might be, and the program will kind of run all the way through any automated steps with minimal user intervention. Our company started a little while ago, technically. So our company was founded as a McGill spinoff in 2010 out of the lab of Nick Montessier at McGill, who was my PhD supervisor. Him and his partner at the time, Eric Terrien, who has now moved on, started this company, translated the academic research into this commercial entity. They did this to the best of their ability, keeping the company going for 8 years, while focusing on some of their other priorities. Nick is still a full-time prof at McGill, for example. We started kind of full-time business operations in late 2018. That coincided with us opening our first dedicated office, which was at what used to be called the Neomed Institute and is now called the Admiral Bioinnovation Center here in Montreal. Admiral was formed, I think, just about a year and a half ago, and is meant to be a pan-Canadian facilitator, I guess, of translating academic research and commercializing companies here in Canada. Our ecosystem is flourishing. You may have seen some of the recent IPOs for, Repair, or Upseller has been in the news for their work on COVID. Yeah, things are really fun here, except for maybe the weather, where I think today it's -20 Celsius. Our team is growing also. When we started in October 2018, it was just Nick and I. We've bootstrapped our growth to now hire a business development manager, Jeff, and bring on 2 application scientists. We announced Mihai joined us as part of a MyTax fellowship, which is a Canadian publicly funded opportunity to help postdocs kind of translate to the industry side of things and do some time in academia for their postdoc and sometimes with a company, and then join full time for hopefully their career. We have 1 more application scientist that we have an approved project, but it's yet to be announced. So stay tuned. Hopefully sooner rather than later. We're having some border troubles because of the pandemic. We work with companies around the world. That's one of the advantages to being in the computational space. We have either software licenses or industrial contracts in over 30 countries on 6 continents. So really working globally. We have for our history, over 300 academic licenses. I think today there are over 60 or 70 active academic groups using our tools in their research. You can see some of the usage cases and references on our website. And we've partnered with over 30 companies. A lot of that being in the last kind of year and a half as we really aim to grow our company. So some context. I don't know everybody's background in the audience. I'm going to focus a bit the context on drug discovery, just to tell you kind of why we do what we do. So drug discovery is really hard. You may have seen versions of this kind of pipeline. You have target identification. Then once you have a target that's relevant to your disease, you do some screening and discovery usually. Then you do some optimization of those molecules into what are called leads, and then you optimize those for different pharmacokinetic properties to make sure that they're absorbed, that they're not metabolized too fast or too slow. And then you have some kind of regulatory clinical trials. So here's just some examples. This is what we look at all day: proteins. Here's some molecules, doing some more advanced assays, maybe making sure the PK, as I said. And then here we have Health Canada in the US, the FDA. There are organizations all around the world that do the regulatory side. This process is extremely expensive. I'm sure that, again, you've heard of this plenty of times. Basically, all in all, the whole process, depending who you ask, is somewhere between 15 to 20 years when you account for all the failures, obviously. And the cost ends up being close to $1.5 to $2 billion these days. This is kind of in the siloed pharma model where lots of projects don't pan out, but obviously you spend money on them until you know that. We focus our research kind of in this box here, or maybe in these 3 boxes a little bit, and we try and provide software to address the needs in those 3 areas. So industry experts believe that about 30% of the time and cost spent in those early stages can be saved using the appropriate, I'll qualify this, the appropriate CAD technique, CAD, computer-aided drug design. These are, CAD is a tool, an analysis tool, a prediction tool, the same way that we use MS, mass spec, or NMR. These are tools to help analyze and accelerate research, and you have to use the right hammer for the right nail, so to speak. So what are the hammers that we've put together? Some of the software, and I'll go into more detail on each one of these in the coming slides, but just to give you a broad overview, we have tools in structure-based design. This is kind of the flagship software of our company. It's called Fitted. It's well documented, published over the last 10 to 15 years. What really sets it apart is some of the more niche, I guess, applications like covalent drug design, metalloenzymes. We'll have some things coming out about nucleic acids. And so this is, we have specific focuses on those areas. We really drive our development by experimental need, not just by what's necessarily out there. This approach has pros and cons, but it really leads us to address the unmet needs of the moment, which tend to be, the unmet needs of the next moment as well for somebody else. So that's what led to the design of these, and I'll give some examples in just a moment. We have tools to address some metabolic liabilities. So this is more in the lead optimization stage, less as a screening tool. So our program is called Impacts. We predict the site of metabolism by CYPs. So cytochrome P450s are responsible for, I think, something like somewhere between 75 and 90%, I think, of first pass metabolism of any xenobiotics, so any drugs. So knowing their metabolic profile allows you to discern if there are chances for toxicities. It doesn't exactly correlate to quantifying half-lives, which is a question that we get very often. That's something else that would be under development, but this just predicts the oxidation profile, and then you can play with it, with your molecule. Again, I'll give some more details. We also aim to predict CYP inhibition, CYP induction as well, which are liable for drug-drug interactions. As chemists, we also have interest in the kind of the more chemistry side of things, which is where Chemspace has been a huge help. We have a lot of tools in chemoinformatics, doing library design, library cleaning. We have substructure search, combinatorial chemistry, where you can have user-defined chemical reactions. We really come at this from the chemistry standpoint. These are not enumerations. They take into account rings, symmetry, stereochemistry, protecting group and leaving group chemistry. You don't want to, if you search a catalog where you can have 5 different protecting groups and you kind of enumerate that in a combinatorial way, you might end up with 1,000 of the same product, because all those protecting leaving groups go, and you end up with that core scaffold being the same across the board. So we take that into account to not overproduce virtual libraries, and then that becomes a burden if you do any virtual screening. We also have tools to do any clustering and chemical similarity. Again, this is a way to kind of automate a lot of the processes or the projects that we work on, where this can all be done kind of seamlessly in a workflow type of setting. And the last thing is small molecule catalyst design. So this has more application in process chemistry, it's in designing new efficient syntheses for hard to access drugs potentially. This is more, I guess, of a novel application of comp chem. Yes, there's a lot of computer-aided organic synthesis. I don't mean to downplay tons of contributions in the field, but this more advanced or higher level type of prediction is more novel. So it comes with the burden of education and understanding what the tools can and can't do. But we just had a Nature Catalysis paper come out a few months ago that details some of the progress we've had, and I'll talk a little bit about it here, some of the projects that we've worked on. So focusing on structure-based design. So I'm going to talk here about, for those that don't know, interaction between ligand and protein. So you can think about it as a key going into a lock. You can think about it as a triangle going into a Pac-Man mouth. We try and best model how a small molecule will fit inside of a 3D structure of a protein. There are a couple of different ways to do that. I'm going to focus on docking. You may have heard of de novo design, where you grow a molecule within a protein, or free energy perturbation, which is a higher accuracy, for quantifying small changes in molecules and how they bind. You would use that technique. Again, here, one technique does not solve all problems. Here's a little graphical representation of, here's your protein, here's a small molecule, and we want to put this, properly within the protein and model how it might go. The idea behind this technology is to provide a rank ordered list where you have, let's say a list of molecules represented by these red lines. You know that the actives, so the molecules you actually want to test in the lab, would be these green lines. So at first it's kind of all mishkabibbled together. What you want to do is dock all these molecules, score the different poses. So that's the two elements of docking, is the sampling of the conformational space and then the scoring. So being able to assign a value to which one is better and which one is worse. And then you want to rank them. And then you hope at the end of the day, you get at the top of your list all the hits and at the bottom all the garbage. In Brian Schweikit's lab at UCSF, we always call this bubbling up. Bubbling is what you hope to accomplish here. This methodology is obviously constrained by protein structure. There are ways to generate structures, but in any CompChem technique, the more variables you add in, the more problems you're likely to encounter, or the more uncertainty, let's say, in your prediction. So what are the things within fitted? I touched on some of them, but here I'll give you some papers that you can go check out. So we have a way to account for some protein flexibility. This was done in the context of base 1 inhibitors. Again, you see here the age of some of these publications. Our software has been in development for a long time and continually improved. We have a way to account for displaceable waters. So using crystallographic waters or waters that you place there, these can be considered within the docking as displaceable. So you let the software decide whether your small molecules should move them or interact with them or stay away from them. But you can keep a fully hydrated site. Full disclosure, there are some cases where this is super crucial, and there are other times where we don't necessarily see an improvement in the results that of having water there or not. I think that most people in structure-based design would attest to that. I mentioned covalent docking. This is a cornerstone of fitted. Being able to simultaneously screen non-covalent and covalent molecules within the same library, letting the software algorithm decide whether a covalent bond should be formed or not, and being able to do this within one run. It means no pre-preparation of libraries. It means no identification, manual identification of warheads. All you need to do is signal to the software, I want to target this amino acid, and you can force it to be covalent, or you can allow it to decide on its own and do a simultaneous non-covalent covalent screening. Metal coordination. So here it was in the context of discovering HDAC inhibitors, which are zinc-containing metalloenzymes. We encoded, this was part of my PhD work actually, we encoded the proton transfer that happens. So this is a biochemical process where hydroxamic acids are known zinc binding groups. As the hydroxamic acid approaches the zinc metal, the proton that was on this oxygen here tends to get shifted over to a neighboring basic residue. And in almost every zinc metalloenzyme that we came across, there's either glutamic acid, aspartic acid, or histidine that abstracts this hydrogen. And so as this moves over, this binding becomes stronger. And so modeling that process is something that we encoded in, and displacing the water molecule that is normally solvating this zinc. We're now working on some additional metals that have come up for relevant projects, and that will be published soon. And last, as I mentioned, nucleic acids. It's become a growing interest to design small molecules targeting RNA and DNA. There was a recent FDA approval. There's obviously aminoglycosides from days of yore. Right now, we just published a methodology placing important water molecules that are key for binding within nucleic acids, key to the modeling. If you've worked with proteins versus nucleic acids, you know that today's force fields are not great. The polarizability within nucleic acids, the charges, protein force fields are not adequate. So we've had to make some adjustments, and we hope to have a docking study published soon on what we've been able to achieve. So how does fitted actually perform, I guess, on some larger studies? So we like to point to third-party studies that we don't have a part in. That way there's no specific tinkering of any parameters. You know, sometimes there's a secret sauce. Certainly when we do a project as a service, we add in our secret sauce. But from a software, you know, purely software standpoint, it's nice to have somebody else test it for you where you're not involved. So I'll point to two. One is from a little while ago at this point. This was a study out of Australia where they tested, I believe it was 6 targets, including some nuclear hormone receptors, some kinases, and some other enzymes. And this is not, I don't point to these to say we're the best or that anybody's the worst. Every protein and every project can be different. I acknowledge that. I hope others acknowledge that. But we do perform near the top, which is what I think is important. You know, here these are sums of Pearson correlation and Spearman correlation coefficients, and we perform quite well compared to some of the, I guess, more well-known docking softwares, or at least historically well-known ones. Second is a covalent docking study. So what I was just talking about out of Hungary in Georgi Casero's group, where again we perform quite well. So we're the dark blue guys here. So there's different metrics for success. The bottom part here for everybody is, I think, the best out of 3 runs. The lighter is the best out of 10 runs. And I'm happy to point out that, I mean, they were very generous with us in terms of some covalent groups that we had not encoded. So we hadn't come across these groups in our projects. We've now, since the time that this paper came out, we've been working on encoding these new groups and running a wider benchmark set. And that also will be published hopefully in the next, let's say, 2 months. If the team is on the call, then I hope they heard that 2 months. But again, the point here is not that someone is the best or the worst, but that we perform quite well with some of the world-renowned players in the field. So here's the first case study that I'll point to. This was done at McGill over the last decade, about discovering prolyl oligopeptidase inhibitors. This was done in Nick's lab. The idea was to find covalent inhibitors. This has, I think, a couple of disease indications through 3 design and experiment cycles, always coming back to the structure-based design. They were able to find really, really potent molecules. So we started out with this kind of scaffold. And when I say we, I mean the people in Nick's lab. So credit to them 100%. It started out with this 200 nanomolar hit, then through some modifications, ended up going lower and lower. And today this project has actually reached the patent stage, actually with a new covalent warhead in these boronic acids. And you can go read about this in a recent European JMed Chem, where they're also looking at another enzyme, FAP, which is relevant for the disease indication that they're talking about. Another project, and this is more from the business side and less of an academic project, discovering antagonists for GPCR. So this comes from a client. I'm sorry that I thought we were going to have our preprint on this material out already, and that's why I wanted to include it. But I can't show too many specifics on this just yet. It will be published, so you will be able to see exactly what I'm talking about. But for now, I'll just say that a client came to us, and this is fairly typical for the way we operate. They were looking for some CAD support to discover new GPCR antagonists. The GPCR is a fairly well-known target with known chemistry, which is the ideal case. Remember I said that there's the right tool for the right project. This is where CompChem thrives. GPCRs are also, they have their challenges, obviously, but from a structure-based design standpoint, there's a lot of successes, and I can point to all the research out of Brian's lab and his collaborators on GPCR specifically. The well-known chemistry that's in the literature and well-documented allows us to properly benchmark, make any of those small, tinkering adjustments that we want to really enhance the predictivity, and then go forward in a prospective way. So this project is more recent, and after our, we came into contact with Yurii and Chemspace, And so we started from their library of 5.3 million compounds. We applied some filters that were important for this target, in terms of some key warheads, let's call them. This isn't covalent design, but we'll call them warheads. Some other key chemical properties. We docked those 200,000 molecules. We had some optional filters that we applied based on the retrospective modeling, where we knew that, let's say, an interaction with residue X was important. So we can capture these things from the docking in an automated way, and then just filter out the ones that don't make those interactions. We then clustered by similarity after, so that we didn't look at just redundant analogs in the first go. We visualized about 200 of the poses. We selected near 50, and we had about a 20% hit rate. Hit rate in this case defined as it was either 10 or 30 micromolar. 30 might have been generous. I think that we had 9 under 10 micromolar, which is pretty good out of docking. I can't show you structures just yet, but all of these were obtained in dose response. So I hope that this makes you believe me, essentially, that these are not false hits. These are functional assays, and we have some follow-up being done in terms of some SAR and potentially some actually binding assays to ensure that they're doing what we think they're doing. But 160 nanomolar right out of docking is pretty cool. We haven't done any kind of med chem on those to improve them, and so we'll see where the project goes. But this was exciting for us. COVID-19, everybody and their grandmother, I think, has worked on COVID-19. Similarly, at McGill, we dove in, but we didn't want to just do a computational study and put that out. For those that follow the literature, I think you know that there was plenty of that. And there was a focus by Nick and his group to really focus on experimental testing. You need experimental testing, otherwise your model is just a model. So what they started on back in, I guess March is probably when they started, was looking at the main protease. It had structures. It was fairly easy to work with for them, especially in terms of covalent design. So they took this known non-covalent inhibitor, and the goal was to turn it into a covalent binder with these proximities in mind. And so using our tools, they designed a virtual library. So breaking this on, I think there was 2 approaches. One of them was breaking it here and then adding on groups to the nitrogen, and one of them was just breaking off this imidazole and putting on groups here. And so from the 2 approaches came some hits. They bought reagents. Sorry, they synthesized and tested the molecules in assays, and they got, this was, I think, one of the better ones, 400 nanomolar binder to the main protease. Since then, they've also applied this to PL-Pro, the other protease, papain-like protease, and that project was done. We supported that project a little bit here at MFI. And that's sourcing commercially available compounds from Chemspace as well. So Yurii and his team, Olga, Rita, have been a huge help for us in this area. The worst thing for a comp chemist is to screen compounds, really like compounds, and then not be able to get them. It's pretty demoralizing. So we've appreciated the support from them. Moving on a little bit, so metabolic liabilities, this was our impacts, as I mentioned. So I'll show you quickly a bit how it works and then give you a quick case study as well. So you take a 2D structure of a small molecule. This is a molecule that you've taken, it hits, this is flurbiprofen, but let's say any molecule hits your target of interest. You want to further study its metabolism. So the software will automatically identify different reactive sites for oxidation by CYPs. We combine some of our structure-based design approaches to orient the molecule within the cytochrome P450. We have the 7 major CYPs are encoded already by default that you can use. They're ready to go. So all you need is the small molecule. We use the structure-based design tool and some transition state modeling to predict the 3D conformation of that oxidation reaction. Why this is important is it allows you to make adjustments to the molecule that may not be just at the site of oxidation. a typical strategy is, okay, well, I'll fluorinate the site that's oxidized. However, if that site is crucial to the recognition of your protein and the potency that you see, then what this 3D image allows you to do is be a little more creative and make adjustments elsewhere that might disrupt this pose binding within the cytochrome P450 and change the metabolism profile. And we have a partner in pharma that has done just that, and it's really cool. They've actually upped and lowered the metabolism of different sites on different molecules. Overall, this is a little bit dated, so I think that there's a new update coming soon. But the standard metric in this field is you predict 2 sites of metabolism. If either one of those 2 is correct, then you say that's a successful prediction. So in previous benchmark that we had with a data set of manually curated molecules of over 700, we had between 75 and 80% across the board. CYP3A4, I think, notoriously lowers that a little bit because it's such a vast binding site. And so sometimes the structure-based component has a bit of noise to it versus 1A2, that's a pretty well-defined binding site. And so the structure-based component is a huge help in that regard. Other things that are being worked on in impacts, and they're kind of still in beta, I guess, is predicting CYP inhibition. I mentioned this, so coordinating the iron directly, that's at the center of the heme in the CYP. CYP induction, so primarily we're focusing on binding to PXR, which induces CYP3A4 expression. And last, and has been notoriously a challenge, is CYP selectivity. This is kind of getting more towards individualized medicine where somebody might have a higher expression of one CYP versus the other, and so being able to predict that may reduce some toxicity liabilities that usually are only illuminated in later stage clinical trials, and therefore are a huge time and cash burden for the R&D teams and the pharmas working on them, or biotechs. Here's a case study of how this can be put in practice. This one is also a little bit older. It comes from the earlier days of Molecular Forecaster, working with AstraZeneca on this project. It shows how the tool is meant to be complementary and not a screening tool necessarily. This was a molecule that they were looking at, and you can see it shares similarity to some of the POP molecules that we were just talking about. And this was kind of the oxidation profile that was coming out of their software or analytical tools. And so the somewhat obvious prediction getting 3 peaks is that, well, you have, ortho metapara on this ring, and that's where this molecule is being oxidized. However, it was observed that this molecule was basically not CYP oxidized, or certainly wasn't the same profile at all. And if you notice, the only change here is the sulfur for this oxygen. It's not something you'd expect to see if the oxidation was actually happening on this ring over here. AstraZeneca got together with the lab at McGill, and Impacts was actually predicting, based on the 3D pose, that it was actually the sulfur in this ring that was being oxidized. And it turned out, after some extensive work, that the sulfur is indeed oxidized. It results in a ring opening here that then results in all kinds of scrambling of these stereo centers, which leads to different profile, different ring closures, ends up with different products, and then these can be separated actually. So it ended up being a pretty large project, did a lot of work on it. It was a pretty cool comparison or demonstration, I guess, of how software and experts can work together, which is what we hope, I think, for all of these tools. Switching gears entirely, I'm going to take a small drink of water, towards more of the organic chemistry side of things, or medchem. We have our suite that we call Virtual Chemist, because it's meant to think like a chemist, act like a chemist, help a chemist, basically. So I'm going to focus on the asymmetric catalysis part of this. We took our the team's expertise in chemistry and try to incorporate it into one of our software modules. So the first thing is the Hammond-Leffler postulate that says that the transition state of a catalyzed reaction looks most like the species that it's closest to in energy. So here's an example. And you can make this into a mathematical equation. I don't want to show too much math, but the forming bonds can be a linear combination of the bonds being there and the non-bonds being there. The second important factor here is that we can compare energies that go towards one enantiomer or the other, and we can do this with molecular mechanics this way, rather than using higher levels of theory, which are obviously more time costly and resource expensive. So I'm skipping a lot. I suggest you go back to some of those papers if you want some of the details. But hand waving, ta-da, we end up with a project that we tried. How the molecular mechanics based prediction compares to some DFT, which is obviously not the highest you can go in theory, but is certainly more advanced and usually amenable or viewed in the literature as a decent approach. And here's an example. This was different catalysts for, I believe, an epoxidation reaction and Diels-Alder, maybe. I don't remember exactly all the details. The point here was that the tools were pretty comparable in predicting EEs. I know some people are not fans of EEs. You could show this in terms of energies. The mean unsigned error is usually around 1 or lower than 1 kcal per mole, which is actually pretty good. But this is a nice way to kind of show how these are somewhat similar. So that leads me to a case study, how you might use this type of tool. The same way that we try and screen small molecules binding to proteins in drug discovery, we want to know, can we screen small molecules as catalysts in different reactions? Catalyst being likened to a drug and a reaction or a transition state being likened to a protein. So similarly, you assemble a list of known catalysts and decoys or known misses, represented again as these kind of red lines and green lines. You want to compute the stereoselectivity and rank them. If we can quantify, it's even better, but if we can just classify that the best are at the top and bubble them up, then that's a success as well. So you compute these and then you evaluate how much you've enriched the top of the list for these stereoselective molecules. So how does this actually perform? So one example that you can find in the Nature Catalysis paper is a sheet epoxidation. We took 16 known catalysts and a bunch of decoys, just other chiral ketones. And you can see here, if you're familiar with looking at kind of an AUROC type of plot, as you find more hits, this goes up. In the ideal scenario, you go straight up and then across, basically. And the area under this becomes 1. This does pretty well. It finds a lot of the most selective. You can see also in this representation that you find a lot of these hits right at the top, and there's the selective catalysts are highlighted basically by the software that then you could go test in the lab. Similarly, this was done with another reaction, Diels-Alder reaction. So we took 6 knowns and a bunch of other chiral amines and tried to predict and rank the stereoselectivity. And similarly, it performs quite well. We end up with half of them in the first, 5 to 10% of the list, which is what you want. You just basically don't want to test in the lab all this stuff. It takes a really long time. I encourage you to go check out the paper. There's more. I'm not doing it justice, but I also don't want to sit and lecture you all for too long. The last kind of case study that we thought about for this is optimizing a catalyst. So this is one where you have a good scaffold and can you make it better and make it more stereoselective. So we took this really neat study from a group in Italy where they did a ton of work, huge credit to them. This was all done experimentally where they made their own kind of precursor. They used this to make their catalysts over here. And then they used those catalysts in this Diels-Alder reaction here, making different products. And so if you think about what actually needs to be done here and how a computer might be able to reproduce it, you need to identify and purchase the right chemicals that fit these scaffolds. You need to make this library. Then you need to make this library. So you prepare and you have to separate them in the lab. Obviously, that's not trivial at all. And then you apply all these catalysts. And there's some cool robots that can accelerate that a little bit, but this takes a really long time to do. I'm sure that this project took this group, if not 6 months, then a year, and if not a year, then 2 or 3 years. What we did was we built a workflow. This is a quick look at our software, how you can build these kind of custom workflows to do everything. I'll kind of overlay what each kind of section of this workflow does. So one of them will look for the substructures that fit this criteria with the right leaving groups. Then we'll do the CombiChem to make a unique list of these, so no redundancies. We'll then do the second reaction to make all the catalysts. So 1, 2, 3, we'll make all these. And then we'll build a 3D transition state and test them and predict the stereoselectivity of each one. Thankfully, this group in Italy did all that work, and so we had experimental values to compare to, which is quite rare and an extensive amount of work. So can we prioritize the selective catalysts? First, we need to build, like I said, these 3D transition states. We do this based off of templates. So getting some of the core pieces in place, and then we can kind of template all the different substrates and catalysts onto this template. And then we predict stereoselectivity. And similar to the other plot that I showed, you have predicted EE here on the Y, observed EE on the X. The quantifying is difficult, but what I can tell you is similar with docking to drug design. It's not so important in the early stages of this to necessarily quantify. You want to highlight the selective catalysts, and then you can optimize a little bit more in the lab after. So this box here in the middle being basically uninteresting catalysts that are not selective at all. What we really want is to focus on these extremes, and we capture a lot of them. We obviously have a couple of outliers that we miss, but you get a really good enrichment of these selective catalysts here. And so now we're applying this in kind of a prospective way to try and really hammer home that, you know, virtually you can do this much quicker than running experiments on all these and the cost associated with it. So wrapping it all up, what does that all mean for you? Hopefully you like the science, but hopefully you also can benefit from our support. So we work in generally 2 ways. One of them is through contract research. So we can do this on a project basis, you know, in a fee-for-service model. We also have some FTE-based clients where we provide all the support that we possibly can. We like to design our projects with go, no-go stages. Again, if you can't retrospectively model available data, then the odds of success going forward are slim. We don't want to waste your time. We don't want to waste your money. We'll work those into our proposals. The other way is to license our software directly. So we charge an annual fee. We can put modular packages together if you're interested in drug discovery, but not in catalyst design or vice versa. We don't charge you for everything. We're pretty flexible. And last, we've been looking at options in incorporating our algorithms into more extensive visualization packages, such as Samson from 1 Angstrom. The integration is not fully complete yet. We don't have everything available. But these are things that we'd be willing to discuss with anybody interested. Working with us is, I like to think, and I'm told, pretty easy and an advantage to those that come to us. We have high accuracy tools, which means you get really good quality data. Our team is diverse with expertise in quantum mechanics, structure-based design, chemoinformatics, medicinal chemistry, and beyond. We all contribute to projects when it's necessary. So you get a lot of perspectives and a lot of insight to your work. We use our own software, which means that we can modify, adjust. If there's issues, we don't have to worry about downtime, don't have to worry about third-party licenses and agreements and things like that. We don't ask for any IP in projects. So if you're an early stage biotech or you have a young idea you want to check out, there's no risk to working with us. You're not parting with the most valuable part of your work. We don't develop our own internal assets, so that means that our best talent is applied to your project and not in-house to our own. And we have these awesome strategic partnerships with companies like Chemspace, which means that molecules are accessible, and we can really connect you with the sources for those work. We have friends in crystallography and other supporting tools. It really helps in this kind of broken down or deconstructed, let's say, pipeline of drug discovery and research. So if your software usage has gaps, there's certain interests that you have that are not being met by your current tools, we're happy to try and complement what you have. If you have CAD users and the pipeline is just too big and you need some extra hands on deck, or if you have none at all and you want to know, maybe is CAD amenable to your project, we do all this kind of diligence ahead of signing any agreements, well, except maybe a CDA. So that way we can offer you our best advice on whether we can support you or not. And for the last thing, for our ACE project, if you're interested in some of that, that's a little bit of a new frontier, and it's more collaborative research. We have a few cool partners like Liverpool Chirochem that we're working with to try and apply this prospectively to get to some new cool chemistry. Then we'd be happy to talk with you. Talking is always free. So I appreciate everyone sticking it out for the hour. I hope I didn't step into your lunchtime, certainly here on the East Coast, or your breakfast time on the West Coast, or your dinner time in Europe. Please reach out to me. You can visit our website. It has a lot of details. And I'm happy to answer any questions now or any questions you have later.
Thank you, Josh, for this really nice presentation. So we have a few questions, and one is in the segment of catalyst design, does your algorithm fit rational design of enzymes or peptides as catalysts, or it's strictly directed towards transitional metals?
So it's not just metals. We do organocatalyzed reactions as well, but what I've shown here is primarily directed at small molecule, what we would call small molecule catalysis. We have had some exploration in the past into biocatalysis and working with bigger macromolecules, but there's nothing. It would be more of a research project to port this technology. There's not necessarily a reason to think it wouldn't work, but we don't have a specific tool for biocatalysis just yet.
Thank you. I have a question. So have you thought on integrating your tools with a system for automatization like KNIME or Pipeline Pilot?
We've talked about it. We've always just generally from a, I don't want to say a policy standpoint, but from a philosophy standpoint, tried to keep everything self-contained. Not because we don't want to work with others, not at all, but just because then we have the access to anything that's buggy or broken. It's within our control, and we can fix it. The important thing to know for our software, if you were to use in-house, is that the user interface is really just a front end on a back end of coding. So that back end can be integrated into any kind of script. The inputs and outputs are standard formats. What changes is internal to the modules. So the way that our ligands and proteins are prepared for docking and things like that is internal. So we take standard formats, we output standard formats, and so those files can be brought in and out from any other software tool, whether it's something like KNIME or something like something internal to a bigger pharma.
Thank you. So I think we don't have any other questions. So with that, I would like to thank you, Josh, and everyone for joining the webinar. So if you have any questions, please reach out to Josh or to myself. And yeah, with that, I would like to close the webinar and thank you. Good one. Bye.
Ciao, Yurii.